Hi students, welcome to PhD Jobs and Admission. This is Gauri. So in today's session, I'm going to teach you about Pondicherry University's PhD Entrance Tests Research Methodology subject. So in this session, we are going to see Research Methodology PYQs of the year 2018 of PhD Entrance Test at Pondicherry University. So from today's session, your concepts relevant to research methodology will get clear. As well, you will get an idea exactly what type of MCQs you have been asked in the research methodology section of the PhD entrance test at Pondicherry University. Okay, so most of the PYQs also get repetitively asked in each year, so that all of you candidates can also revise these MCQs and you can solve these MCQs, and after that obviously you can solve all of these MCQs in the current PhD entrance test which is upcoming. Okay, so now let us begin with this session. So students, as you know that there is PhD entrance test at Pondicherry University, and all the candidates have to give this PhD entrance test for PhD admission. In this PhD entrance test, all the students have to solve MCQs from research methodology and as well MCQs on concerned subject. Okay, so you need to score 50% marks in the PhD entrance test so that you can qualify the PhD entrance test. So students, basically you will be asking 50% MCQs on research methodology and 50% MCQs on concerned subject. So to qualify this PhD entrance test, we provide research methodology complete course. With the help of this course, all the students can guaranteed score more than 45% marks in research methodology section, and after that they can qualify the PhD entrance test at Pondicherry University. In this course, we provide full syllabus video lectures under which theory and MCQ lectures both are available. We provide full syllabus notes and mock tests. We have 10 mock tests. Each mock test contains 40 MCQs. That's why overall you can solve 400 MCQs in mock tests only. By solving the mock test, all the students can understand how much they have prepared for the entrance test and how much preparations they have to do. Okay, we provide more than 1500 MCQs revision PDF. By the use of this revision PDF, all the students can solve all the revision topics in a single PDF, and they will guaranteed get 80% MCQs from this PDF only out of 100% MCQs in the research methodology section of the PhD entrance test. And all of these study materials will be available in Hindi and English, both the languages. So students, if you want to buy this complete course, then you can either download the Global Online app from the Play Store. For that, I have given the link in the description box of this video, or else you can contact me through these WhatsApp numbers. The course fees are only $699. In these low fees, we provide guaranteed selection to all the candidates, and all the candidates for sure can qualify the PhD entrance test through this research methodology revision package. Okay, so now let us begin. Which of these measures can be used to present dispersion of data? So from these options, which is the measures which we can use to present dispersion of data? Now let us see options. Mean, median and mode, standard deviation, IQR means interquartile range and range, median alpha and standard deviation, range, median and deviation. So students, dispersion of data means how much your data scores are spread in the whole data set. So there are different types of measures of dispersion, and those are standard deviation, then variance, then interquartile range and range. That's why here your correct answer should be what? 2. That is standard deviation, interquartile range and range, because all of these are the measures of dispersion of data. Mean, median and mode are the measures of central tendency, which give you the measures of the central position data in the data set. Median is nothing but what? The middle value of the data set. Alpha is nothing but the probability of occurring of type 1 error, and this concept we are going to see in the further part of this session. Standard deviation, range, median and deviation. So if you see other options, in that somehow there is one of the measure of central tendency. Okay, that's why all other options gets eliminated. So your correct answer should be what? 2, that is standard deviation, IQR and range. Now let us see what are the definitions of these measures of dispersion. First of all, there is standard deviation. Standard deviation means how your data points, okay, how your data points are dispersed or deviated from the mean value of the whole data set. Okay, so students, what is variance? The definition of variance is exactly as it is like standard deviation, and whenever you want to calculate the variance, then you need to do the square of the standard deviation. Okay, now let us see what is interquartile range. Whenever we want to calculate the interquartile range, then the definition of interquartile range is that difference between 75th percentile minus 25th percentile. Okay, so how you can calculate this 75th and 25th percentile? In the IQR, what we do, we have whole data set, so we make the 4 quartiles of this data set. Okay, so the data set between 1 to 25 comes under lower quartile, then from 25 to 50 it becomes the middle quartile, then 50 to 75 it becomes what? Upper quartile, like that. Okay, so you make like this 4 parts, and the data which is 75th percentile will be 1 to 75. Okay, and the data which is 25th percentile will be 1 to 25. So you need to what do? You need to calculate the difference between this 75th percentile minus 25th percentile. Then you will get the value of what? IQR. So by getting the value of IQR, you can measure how your data is spreaded in the whole data set. Okay, now what is range? Range means your extreme biggest data point in the data set minus your extreme smallest data point, okay, in the whole data set. Again, range will tell you about how your data is spreaded in your whole data set. Okay, so this is your overall measures of dispersion. Okay, so don't confuse between the IQR and range. Okay, because the definitions of both the concepts are different. Blood test failing to detect the disease it was designed to detect is an example of what? Whenever there is a blood test and this blood test fails to detect the disease it was designed to detect the disease, then this is an example of what? Whether it is example of type 1 error, null hypothesis testing, type 2 error, chance factor. Okay, so students, basically type 1 error and type 2 error are the two types of error that occur in hypothesis testing. So as we know that there is null hypothesis that we test, okay, in the research process. Null hypothesis means that we assume that there is no difference between the variables under study. And there is alternative hypothesis. Alternative hypothesis means we assume that there is difference between the variables. Okay, type 1 error occurs if this null hypothesis is true but still we reject it, and type 2 error occurs when basically null hypothesis is false and still we accept it. Okay, type 2 error occurs. So this example is basically relevant to these types of errors. Okay, because here your null hypothesis would be that patient does not have the disease. This is what actually your null hypothesis, because within null hypothesis we say that there is no difference between the variables. So it means that in this example your null hypothesis should be what? Patient does not have disease. Now you are going to test this null hypothesis, but here you will get that this null hypothesis is false, but still you are accepting it, right? Okay, so that this is an example of what? Type 2 error, because in this example there is a blood test and this blood test is failing to detect the disease it was designed to detect, and that's why here this null hypothesis is false, but still we are accepting it from this example, right? That's why here type 2 error is occurring, because we know that null hypothesis is what? Patient does not have disease, but we know that this is false because here blood test is failing to detect the disease. All of you getting? And still we have to accept this null hypothesis though it is false. That's why this is an example of what? Type 2 error. Okay, this is not an example of type 1 error. Okay, because here null hypothesis is actually false, not true. Okay, chance factor. Now let us see what is chance factor. Chance factor is nothing but an influence that contributes randomly to each observation, and this is unpredictable. Okay, null hypothesis testing is nothing but what? You are testing the null hypothesis by using different statistical techniques. Okay, so students, we provide you Pondicherry University PhD Entrance Test Paper 2 study material as well. By the use of this Paper 2 study material, all the students can guaranteed score more marks in Paper 2, and after that they can qualify the PhD Entrance Test over here. In this study material, we provide full syllabus notes and MCQs. By the use of this study material, all the candidates guaranteed score more marks in Paper 2. Basically, we provide in the MCQ section most expected MCQs and PYQs. By the use of these most expected MCQs and PYQs, all the students can guaranteed score more marks in Paper 2. Paper 2 study material fees are only 999, and in these low fees, we provide guaranteed selection to all the candidates. The subjects that I have shown on this screen, on all of these subjects, we currently provide Paper 2 study material. If students want study material for any other subject, then they can contact me through the comments. Okay? To join our courses, you can contact me through this WhatsApp numbers only. Okay, so students, this Paper 2 study material will be really helpful for your entrance test, basically for your written exam, and as well it is very helpful for your interview sessions. Okay, also because within the interview sessions, examiners will ask you different questions on your research proposal, basically on your concerned subject, and that's why by the use of these full syllabus study notes, all the students can guaranteed study very well about their concerned subject, and they can give the interview very well by using these full syllabus notes. Okay. Plagiarism is, now what is plagiarism? Use of writer's ideas without giving due credit, originality, referring and quoting too many authors, data manipulation. So students, in yesterday's session also we have seen about what is plagiarism. Plagiarism is nothing but whenever we are going to use another writers or authors or researchers ideas and their work in our research work, but we are not giving acknowledgement or credit to them. So this is called as what? Plagiarism. And plagiarism is unethical in research methodology. Whenever any type of a researcher does plagiarism in their whole research process, then it is very unethical, and that kind of research paper does not get published in the journal, and that kind of a researcher have to give somehow the penalty. Okay, so that's why here your correct answer should be what? 1, because plagiarism is nothing but what? Whenever we are using writer's ideas without giving due credit to him or her. Okay, when neither experimenter nor subject knows who is assigned to the experimental and control groups, it is called dash. Okay, so whenever students, we do the experiments, okay, so in that experiments there is one experimenter and others are subjects, right? Subjects means participants. So whenever participants and as well experimenter does not know about the experimental and control groups, then this kind of experiment is called as what? Whether we call it as placebo effect, double blind technique, or type 1 error, or single blind technique. Okay, so students, as we have seen about type 1 error, okay, in previous questions. As we know that type 1 error occurs whenever there is a null hypothesis which is true and though we reject it. But here nothing is happening like that, right? That's why this option gets eliminated. Okay, so students, basically this concept is relevant to the clinical research. Okay, in the clinical research methods, you know, experimenters do lots of experiments on their participants, and there are somehow different concepts relevant to this. Whenever researcher and participants does not know exactly who is there in the experimental group and who is there in the control group, then this type of experiment is called as what? Double blind technique, because both experimenters and subjects don't know who is in the treatment group and who is in the control group. Are you getting? In the single blind technique, experimenter knows who is there in the experimental group and who is there in the control group. But participants, okay, participants does not know. Okay, they does not know who is in the experimental group, who is in the control group until the experiments occur on them. Okay, now what is placebo effect? Now placebo effect occurs when there is an improvement of symptoms is observed despite using non-active treatment. Okay, it occurs due to psychological factors like expectation or classical conditioning. Okay, so students, placebo effect occurs when we don't give any kind of a treatment to the participants, but still participants behaves like, you know, we have given the treatment to them. Okay, fine. For example, whenever we give somehow you know, paining pills to the participants, you know, and they start you know, behaving like they don't have any pain. All of you getting? Okay, here in this situation we give the paining pills, but still they don't behave like we have given them the paining pills. All of you getting? So this is called as what? Placebo effect, which is called as what? Fake treatment effect. Okay, replication of an experiment means, now what is the meaning of replication of an experiment? Reanalyzing the study using renewed design, repeating the original experiment, reporting similar findings, reanalyzing the data using qualitative methods. So students, whenever we do the replication of an experiment, then there are two goals for this. First is that we need to basically verify our previous experimental methods. For the verification purpose, we replicate the experiment, and as well we need to, you know, study about exactly the results that we have got during previous experimental methods are reliable or not. So we need to check whether the results are reliable or authenticate or not, and as well we need to verify our experimental research methods. So for these two purposes we replicate the experiment. So basically we repeat the original experiment to, you know, satisfy these two goals, and that's why here your correct answer should be what? 2, that is repeating the original experiment. Okay, when statistical test should be used with interval or ratio data with multiple dependent variables. So from these statistical tests, which statistical test we can use with interval or ratio data whenever there are multiple dependent variables? So students, interval or ratio data means the data which can quantified. Okay, the data which can be quantified, and within the ratio data there is a characteristic of what? Absolute zero. Okay, so let us see different options. ANCOVA, repeated measures of ANOVA, MANOVA, mixed ANOVA. Now let us see step by step all of these statistical tests, and then we will come to our answer. ANCOVA means what? ANCOVA means analysis of covariance. Okay, analysis of covariance, and this is the extension of ANOVA statistical method. So within the ANCOVA we evaluate whether means of dependent variable, okay, means of dependent variable are equal across levels of one or more categorical independent variable, okay, independent variable, and across one or more continuous variables, okay. So here there is dependent variable, and we are going to analyze or determine whether means of dependent variables are equal to categorical independent variables and as well across continuous variables. So here you can see there is only one dependent variable, there are one or more independent variables, and there are one or more continuous variables. But in the question there are basically multiple dependent variables, right? So we can't do ANCOVA in this example. Now let us see what is repeated measures of ANOVA. Repeated measures of ANOVA means from ANOVA we are getting the repetitively measure. So basically you are repetitively doing ANOVA statistical test. Now the ANOVA is what? Analysis of variance. Analysis of what? Variance. So whenever we need to, see the differences between means of more than two groups, then we can do ANOVA in that situation. Okay, so students, whenever we compare more than two means, then in that situation we does what? ANOVA. Okay, and the full form of ANOVA is that analysis of variance. So basically in this we analyze difference among means, and it splits an observed aggregate variability found inside data set into two parts. One is systematic factors and another one is random factors. So here also there is only one dependent variable, okay, not multiple variables, multiple basically dependent variables. That's why this option gets eliminated. Now what is MANOVA? MANOVA is nothing but what? It is multivariate analysis of variance, and this is an extension of ANOVA, okay, and it allows for analysis of multiple dependent variables simultaneously. Okay, this is very important. In MANOVA it allows us for what? Analysis of multiple dependent variables simultaneously, and exactly this thing they have asked us in this question. That's why here your correct answer should be what? 3, that is MANOVA. Mixed ANOVA, what is this? Mixed ANOVA compare mean differences between groups that have been split on two different types of factor variables, where one factor is what? Within subjects factor, and another factor is what? Between subjects factor. So within mixed ANOVA as well we compare means between groups, okay, basically more than two groups, and here we split these groups based upon two factor of variables types, and from that one factor is what? Within subjects factor, and another one is between subjects factor, okay. But here your correct answer should be what? 3, that is MANOVA. Analysis of variances method is used when? Now whenever we can use analysis of variances method. Analysis of variance means what? ANOVA, like we have seen earlier, okay, in the previous question. When we want to compare two means, when means are independent, when means are dependent, more than two means are compared. So we use ANOVA technique, students, when we want to compare more than two means. Okay, that's why here your correct answer should be what? 4. Whenever we want to compare two means, then in this situation we can use t-test. Whenever the means are independent, then in that situation we can use independent sample t-test. Okay, whenever means are dependent, then in that situation we can use paired sample t-test. Okay, so these are the situations where we can use different statistical techniques. Okay, in testing null hypothesis, if the findings are significant, then null hypothesis is, whenever we are testing for null hypothesis, and after testing the null hypothesis we got the findings which are significant, then what is null hypothesis? Whenever मतलब whenever we can actually do with the null hypothesis, whether we basically reject this null hypothesis or review null hypothesis or accept it or cancel it. So students, whenever we test the null hypothesis and whenever we get the findings significant, then it means that we need to accept the alternative hypothesis. Alternative hypothesis means the hypothesis that we make which is exactly opposite to the null hypothesis, because within the null hypothesis we assume that there is no relationship between the variables, but in the alternative hypothesis we assume that there is difference between the variables. Whenever we get the findings which are significant, then it means that we need to accept the alternative hypothesis. We get the results that there is difference between the variables. Okay, so we are here accepting the alternative hypothesis. It means that we are going to reject the null hypothesis. That's why here your correct answer should be what? One, that is rejected. So whenever we are going to test the null hypothesis and when we get findings significant, then in that situation we are going to reject the null hypothesis. Okay, which one is not a probability sampling method? So from these options, which is the option which is not a probability sampling method? Random sampling, cluster sampling, purposive sampling, stratified sampling. So students, not a probability sampling method is non-random or non-probability sampling technique. Okay, so in this sampling technique we are going to choose the participants non-randomly. Okay, so from these options, purposive sampling is not a probability sampling method, because here researcher has a somehow purpose with the participants, and that's why researcher only select those participants who has those characteristics that researcher wants in his or her study. Okay, that's why purposively here researchers select the participants, not randomly select. Okay, that's why purposive sampling comes under non-random or non-probability sampling technique. Other options like random sampling, cluster sampling and stratified sampling are the examples of probability sampling method in which we are going to select the samples randomly. Okay. Ethnography is a part of. Now what is ethnography? Philosophical research method, quantitative research method, psychological research method or qualitative research method. So students, ethnography is a research method where we study the culture, okay, of a particular society or community, and in this research method researcher uses participant observation technique to select or to collect the data. Okay, within the participant observation, researcher lives with the people in the community and researcher participate with the activities with those people, and along with that researcher observes the participants, and this comes under what? Qualitative research method, because in the qualitative research method we understand, we study how human behaves, how human feels, how human experiences in a particular situation. Okay, so for this we uses what? Qualitative research method, and ethnography is a part of this qualitative research method. Okay, so students, thank you for watching this session. I hope all of you understood everything whatever I have discussed with you in this session. If you have any doubts, you can ask me in the comment box.